We'll go ahead and move back into our Unit 2 review. Wednesday, we're just going to go through and review the quizzes. So deadline for all the quizzes for everybody is before class on Wednesday. And I'll put those in, give you a little bit of extra time to finish those quizzes, but get them in so we can review them. And if you don't finish them by then we'll have to just put it down as a 0. So let that be your warning.
Maybe push them for on Wednesday.
All right, well, here's our Unit 2 review. Finish up with this. Part 2, starting with biotechnology. Sorry, it says Chapter 6 at the top. Of course, that's not Chapter 6. It's just whenever I make these reviews, that's for some reason it just wants to stay on one chapter. At this point, we're into Chapter 9. But trying to get through all of these reviews today, to prepare you for the test, which we plan to have up Wednesday after class. And again, you'll have about a week to take that. It'll be online through Blackboard. And I know it's a little late, but we'll still go ahead and do a midterm, proctored midterm, and we'll work that out next week. So anyway, with biotechnology, as we mentioned early on in the class, we talked about how it's often referred to as GMO, but genetic engineering is a much better term. And there is something that most of the genetic engineered plants have what we call transgenic kind of technology, to where they've had genes from another species of plant and sometimes even other animals put into the crops. And that they do that by using basically a bacteria that uses proteins to transcribe the DNA. It's really a fascinating technology and pretty advanced, really. Amazing that they're able to do that. And of course we have it's kind of crazy to see, and of course this is in 2008. These numbers have gone up quite a bit since then, in the decade since then. Closer to in the 90s for all of these, and closer to like 95% with soybeans, but 86% of the cotton, 80% of corn, and 92% of soybeans in 2008 were genetically engineered to be resistant to herbicides or to insects. They had the Roundup Ready trait, which means that basically you could spray glyphosate on the entire field, and then ideally it would kill just the weeds. Of course, that doesn't always work that way. Some plants were found to be resistant to glyphosate and then develop a natural resistance on their own. And now there are what we call super weeds out there that, at least for glyphosate, are resistant. So on the insect side, they engineered these plants to be able to produce a kind of natural pesticide, a bacteria in the leaves. So if insects fed on the plants, they would eat that bacteria and it would kill them. So, of course, genetic engineering isn't going anywhere. It's only going to increase. And I haven't heard a whole lot about the genetic engineering that they're doing for crops today. But kind of the same stuff, less fertilizer, less herbicide use, which often has good goals, but the execution is not always the best. Let's get into the propagation of plants real quick. And of course, know that plants can be propagated sexually by seed or through asexual propagation methods that are vegetative methods. Know what those asexual methods are and some of the terms associated with them, like the ones pictured in the table here. Of course, vegetative tissue is basically the same genetically. They are clones. There are sometimes that we can have mutations And often plant breeders will actually try to create mutations for plants in vegetative propagation. Many different ways to do that, but they are all basically the same DNA. Steps of seed formation, of course, all seeds have to have three essential parts: a living embryo stored inside the seed, food storage material, which is often in the form of either carbohydrates, proteins, or lipids, and then some sort of seed covering, which controls the dormancy for the plants. For the seed to germinate, of course, first the embryo has to be alive. In terms of viability, it really depends on storage conditions. And ideal storage conditions are low humidities and low temperatures to maintain viability for much longer. We have a thing known as seed half-life, where basically that's for a certain particular period of time for each crop or plant, the amount of time it takes for that germination percentage to be reduced by half. Means that there will always be some that might germinate. Talking about dormancy, know the difference between scarification and stratification. So scarification is where the actual seed coat is altered. And heat treatment and acid scarification are both just different types of scarification that can actually physically alter the seed coat. Stratification, on the other hand, is this cool, moist period that mimics kind of what the seeds go through in their natural climates. They generally have to have some chilling temperatures, some moisture. Of course, they still have to have adequate oxygen, and of course, a time period as well for stratification. And then there are some seed that exhibit double dormancy that might need both stratification and scarification in order to be able to actually germinate. They also, in some cases, have embryo dormancy where the embryo needs a longer period, in some cases multiple years, in order for it to achieve maturity to where it can actually be germinated. So the environmental factors here for germination include moisture, temperature, aeration, oxygen really, in some cases light, and also a freedom from any pathogenic organisms, any particular fungi or bacteria that can cause seedlings to die. Also, toxic amounts of salts can prevent germination. I will say, though, on the organism part, pathogenic, we talk about bad organisms, but there are some cases where we have mycorrhizal associations to help seed to germinate, especially in like orchid seeds. Again, almost all fruit and nut cultivars, many ornamental plants that have been asexually propagated are all clones, meaning that they have the same genetic makeup. However, in some cases we have mutations. We usually call those, when they occur on a single part of the plant, a bud sport. And by propagating those mutated parts of the plants, we can sometimes, make a new variety by actually preserving that particular genotype. Moving on to taxonomy and the classification of plants. Today there are over 500,000, or believed to be 500,000 different types of plants out there. More than 400,000 species, and many different varieties and cultivated varieties as well. And we can categorize plants, of course, by their temperature requirements, whether they're warm season or cool season crops. And on the woody plant side, for foresters, we classify trees into hardwoods and softwoods. which really that all is just going to say that there are multiple classification types for plants. We're not just classifying them based on their physical characteristics, but how we use them too. And those might not be taxonomical classifications that we use, but just more use classifications. Know about the issue with common names and why we stress the use of scientific names. If you look at the peer-reviewed articles like you guys are doing in your crop projects, you see that they rarely use the common names. If they do use the common name, it's associated with the scientific name. That's because, for one reason, it's universal. It's written in a dead language, so the meanings will not change. But also just because common names have so many limitations, because there are too many of them. They can be very confusing because they're so variable, and really they don't serve a scientific purpose. As the example here with jasmine, there are, multiple different species and even genera of plants that are unrelated that are all called jasmine. So as you can see, it could be confusing to use just the common name. Know about Carl von Linné, or Linnaeus, who devised the system for categorizing plants that led to the modern taxonomy of not just of plants, but of all living things and how we organize them. Linnaeus, of course, classified these plants and categorized them by their sexual reproductive parts, their flowers, fruits, seeds. Today they use genomics. They use DNA analysis to actually categorize them. And in many cases, they're saying that Linnaeus was correct. Not always, though. There are definitely errors out there. A taxa is just basically a group, how we base or classify the different plants based on their physical characteristics. Know some of the different phylums of plants, or the phyla, I should say. The plant kingdom, the broadest of classifications, is then broken down into phylums, including the bryophytes, the mosses, and then the vascular plants that include the pterophyta, ferns, and then seeded plants, including cycadophyta, the cycads, ginkgophyta, the ginkgos, coniferophyta, the conifers, and anthophyta, the angiosperms. And generally, we look mostly at the family as what we usually study. So for the purposes for our crop project, you're going to look at the plant's family and then genus and species. And it's important to know the families of plants for identification purposes, but often they also generally have similar requirements for growth as well. Similar temperatures, some similar types of production systems. So knowing the family is important. Know these basic rules of actually writing the nomenclature of plants, that we always capitalize the genus and lowercase for the species. We underlined if handwritten, or italicized if typewritten or printed. Also know that we have different abbreviations such as variety, subspecies, kind of in between there, and as well as the cultivated variety, which are listed in single quotation marks, and all the words in those quotation marks are capitalized. Again, families are grouped together by their similar floral structures, fruits, and seeds, and here's just an example of that in the Solanaceae, the tomato or nightshade family. Know what a dichotomous key is, and I like to kind of think of it as choose your own adventure for plants. There are keys out there that are specific for categories of plants as well as areas of plants too. So for instance, we have a flora of Virginia. It has basically every plant that grows in Virginia, or naturally grows. not crops, but in some cases they do have some crops in there. And it's all this huge dichotomous key that's like 2,000 pages. Pretty rough. Of course, know about the domestication of plants and the centers of origin for particular crops. Germplasm, again, is the protoplasm of reproductive cells containing the units of heredity that we use to basically store in order to keep crops, and well, not just crops, but many different plants preserved. So germplasm can be many different things. It could be seed, it could be the actual protoplasm itself, it could be live plants in some cases too. When we're gathering germplasm, we call them accessions. So if you were to take a propagule of a plant, a gathering event is known as an accession. And of course, the Food and Agriculture Organization branch of the UN is responsible for preserving these accessions, as well as the we have a germplasm repository network in the U.S. that is conducted by the USDA. And you can actually access those accessions through that network. Also know that as many as 1 in 10 plant species is now extinct or endangered, and that number is likely to rise. Also know that we kind of have a reliance on fairly few number of crops. World's peoples are largely fed by only about 20 crops today. So one of the things that we say for food security is to rely on more crops, more broad crops as well, more not just one particular variety or species, but multiple species, ideally. Moving on to energy transfer in plants, photosynthesis and respiration. Know just the importance of carbon and the reason that it has this potential because of its molecular structure to share electrons and create stable bonds. And that's why it is the main element for energy transfer. Basis of a biochemical reaction being electron transfer, and we call those oxidation reduction reactions. Carbon, of course, is the center of this because of its ability to donate and accept electrons. Respiration is the process of converting oxidized carbon to reduced carbon in the form of carbohydrate. And respiration is the process of converting reduced carbon to oxidized carbon. So respiration, of course, involves oxygen. Where does photosynthesis occur? In the chloroplasts in plants. And what does it do? Well, it creates glucose, one of the primary raw materials for other metabolites in the plant that can combine with other molecules to form sucrose, starches, and cellulose. Sucrose being the most common form of sugar transported in plants and also is the sweetener we know as table sugar. Starch and cellulose are both linked chains of glucose molecules. Starch being very important for energy for both plants and humans, and cellulose being very important for fibers as well as just for the structure of plants themselves. Looking at light intensity, talking about how light is intercepted by plants, and just know that light is just electromagnetic energy. And plants have this amazing ability to convert that electromagnetic energy into chemical energy in the form of chlorophyll and some of the carotenoids. And plants absorb all of the pigments, all of the electromagnetic, magnetic spectrum except for green, and they reflect green. We can, of course, grow plants without natural sunlight, but we have to use artificial light. And in order to make artificial light work for plants, we have to have both red and blue wavelengths. Red being more important for floral production, blue being more important for vegetative production. So the lights that we use, sometimes we use one or the other in artificial, like indoor production. But at some point, we have to use a combination of both. We look at the needs of light by plants by intensity. Of course, we have sun-loving and shade-loving plants. Also know that only about 5% of the solar energy, that electromagnetic radiation that actually reaches the Earth's surface is used in photosynthesis. A good portion of it is, reflected or absorbed by soil, so that we have thermal mass. Heat, of course, is good, right? Temperature is a limiting factor for the growth of plants, but up to a point. Excessive heat can cause problems with plants, and can cause such things as just lower crop production, lower quality crops as well. Water, kind of the same deal. You can have not enough, you can have too much. Generally, when we have too little, we get wilt. And basically, wilt is just causing the stomata to close and not allowing gases to escape, which then halts photosynthesis for a period of time. Too much water, though, basically limits the amount of oxygen that you can have around roots, which limits their ability to uptake minerals and also decreases photosynthesis. Water moving through the plants in the form of transpiration works together with photosynthesis. So transpiration is kind of like a key part of the respiration of plants. We have a few different photosynthetic pathways. Most plants go through the C3 photosynthetic pathway, but then some plants, such as those that grow in arid regions, have C4 mechanisms, especially with tropical grasses, which is a little bit more efficient, named C4 because it makes a 4-carbon organic acid. And again, is more efficient. And then also the CAM pathway, which are used for succulents and cacti, which allows them to basically kind of reverse the whole opening of the stomata. Instead of during the day, they open at night. So they lose less water. I also know that there can be sinks and sources of plants, and as a leaf reaches its full size, it becomes a source, because at that point the carbohydrates that are synthesized in the leaf are beyond what the leaf needs, and it's transporting them to other tissues in the plants, such as roots, fruits, or seeds, that are at that point called sink tissues, is moving those carbohydrates to those tissues. Respiration, again, is the extraction of energy stored in glucose and converting it to other forms that are important for the metabolism of plants. And in some ways, respiration is kind of like the reverse of photosynthesis in the way that it moves carbon dioxide and carbohydrates. Know that ATP is the energy currency of all living organisms, and it's transported to other places in the cell so that it can be used for a wide variety of other chemical reactions. Net photosynthesis is the term for the amount of photosynthates produced above the amount required by the plant itself that are then fixed into biomass, which is kind of looked at as the productivity of plants and ecosystems altogether. Of course, some plant material will continue to respire after things are harvested, and that can basically shorten storage life and lower the quality of the products. We want to try to limit respiration if we can. And one way that we do that for apples, for instance, is by waxing them. Let's talk about water. 90% of most of the plant's mass. For most plants, some cases it's much more. It's required for seed germination. It transports photosynthate, and biochemicals, and it cools the plants through evapotranspiration. Without water, as you all know, plant growth and development will be inhibited and eventually cause the plant to die. Some characteristics of water that are important to know is, one, that it's a universal solvent, which basically allows it to move those photosynthates throughout the plant, as well as move nutrients through the soil. The polarity of the molecule is one of the things that creates the unique properties that water has. One of those being surface tension, which is very important for holding water to particles in the soil and allowing it to move towards the roots. That's in surface tension, we have cohesion, these water molecules strongly sticking together. Of course, water also has 3 phases. It's unlike most other molecules in that it can occur as a solid, liquid, and as a gas. It also has very high specific heat, which allows it to store heat without changing too much in temperature. And that's important for all life on Earth, really. As water freezes, it kind of makes more of an open, porous structure. And just know the different phases and how they look on a molecular level. Of course, when they're solid, they have a very strong, rigid structure that is still open and porous. In the water stage, they're kind of just moving around randomly. And then in gas, there are fewer molecules moving very quickly. Water moves through plants and carries mineral nutrients and oxygen. As well as moving through the soil, it does basically the same thing. It carries the mineral nutrients. When a soil's pore spaces are filled with water, then that soil becomes saturated. And again, that can be unhealthy for plants if it is saturated for too long. When the pore spaces are filled mostly with air, the soil is too dry for good plant growth. And the ability of a soil to retain water is its water holding capacity. 3 forces that cause water to move within soil include gravity, which is just that downward movement, adhesion, which is water particles being attracted to solid particles of soil, and then cohesion, which is water bonded with water. And the forces of adhesion and cohesion together allow for capillarity, which can be the movement of water in pretty much any direction, even upwards against gravity. And here's just kind of a depiction of capillary rise occurring. And of course, the adhesion forces, especially sticking to the particles of soil, makes it difficult for plants to actually extract the water. The forces that cause water to move and that energy that is combined there is called water potential, and water potential determines how water flows. And always water is going to move from an area of high energy to low energy, from wet soil to dry soil along this kind of wetting front. And there's different energy there that a soil or really roots in the soil, must overcome in order to be able to extract that water from the soil particles. When soil is saturated and there's 100% relative humidity in the air above, then water potential is 0. And that allows it to be strongly moved by gravity. As we irrigate, water tends to kind of move in kind of this teardrop shape, can move upward and laterally through matrix potential, resulting in the attraction between the soil particles themselves and the water molecules to each other. Again, those forces of adhesion and cohesion. Salts are often present in soil water, especially in the form of fertilizers. And when there are salts present, they create osmotic energy, which can prevent water movement into the plant, which is negative, right? So overuse of fertilizers can cause a accumulation of salt, which can basically draw water out of roots and then cause up, on the tops of plants, what appears to be sort of a fertilizer burn. So in picture A there, we've got a saturated soil, and then B is after the gravitational forces have removed water from saturation. That's what we call filled capacity. When drainage ceases, but those forces of adhesion and cohesion are still holding water towards the soil particles. Once it continues to dry, though, that water potential decreases, so it requires greater energy for plants to be able to actually extract water, and in C there, we could get wilting of plants. Adsorbed water is hygroscopic water. It has this really high energy, basically, that the plants have to overcome in order to be able to actually extract that water. And really hygroscopic water, that's when we get temporary wilt, and really when there's too much energy for plants to be able to overcome, so plants cannot use that water. So available water is the water that is held between filled capacity and the wilting point. So it's when the water potential is high enough to where plants can overcome that ability. So really the gravitational water is not really available because it's still moving downwards. It's once it reaches fill capacity, after all that water is drained off, that plants are actually able to absorb water. So if you see a plant wilting, then basically it's hit the temporary wilt point. And when you add water, you'll quickly see, especially in a very dry soil, you'll quickly see all of that water, or most of it, begin to drain off. And there's a period of time there. After that water drains, then the plant is able to actually extract water and then become turgid again. And that is that period of time, between the wilting point and filled capacity, in which they're actually able to extract that water. And of course, different soils, different textures, and soil media components can cause that to be quicker or slower. So water actually enters the root's xylem through active and passive forces. And then we have the upward movement of water through the xylem. And that also carries the dissolved minerals that are used for plant nutrition. Moves it to the actively growing parts of the plant. Water flow from the roots to those uppermost leaves and throughout those leaves are known as the transpiration stream. And that is what gives plants their turgidity, as well as regulating the temperature of plants at the leaf level. Mentioned osmotic energy, but osmosis itself is just the flow or diffusion it takes place through a semi-permeable membrane. So it's a little bit different than that salt example. Osmosis does occur in cells of plants through a concentration gradient difference that allows these flow conditions to occur. Again, turgor pressure is what gives the plant its rigidity, and that's that transpiration stream. Sugars and other foods move primarily through the phloem tissues in the plants, which can be upward and downward, as well as lateral too. Nutrients have some sort of barrier to be taken into the actual cells of the plants. And most plant nutrients have a particular system that allows the nutrient to move through them. So particular proteins especially are involved for moving particular nutrients. We consider a plant nutrient to be the charged form, the ionic form of the elements within plants. And some of these ions can have different charges and different chemical forms that can make them more or less available. So know what some of these forms are, especially in the case of like nitrogen and phosphorus and sulfur as well. These are three major macronutrients that do not occur in the elemental form or are not able to be uptaken in the elemental form. Potassium, on the other hand, is. Again, xylem and phloem, moving water and nutrients through the plant. The phloem actually extends a little bit longer, as you can see in this picture, than the xylem does, because it has to deliver nutrients to the growing root as well. And it's often the root hairs that are actually uptaking nutrients, but we have the meristematic zone down here. So the phloem has to be able to get those carbohydrates down here to that rapid cell expansion area. The Casparian strips are these waxy deposits here in cell walls that prevent the movement of water and ions into the xylem, and has to be overcome by some sort of nutrient transport system. Cell walls of plants are composed of both cellulose and hemicellulose, as well as pectin, but the cell wall itself is porous, and nutrients can be passed through the cell walls through these spaces in the cellulose fibers. So we have apoplastic movement and symplastic movement, which are both important to be able to get nutrients actually into the plants. There's an endodermis there that is composed of cells with walls impregnated with this waxy substance known as suberin, which creates an impenetrable barrier again for water and ions and forces them to pass through the plasma membrane or plasma lamella, and that is known as symplastic movement. So how do they get through that plasma membrane? Through these proteins that are embedded in the membrane that serve as channels or carriers for the ions. Another important part in plant nutrition is that some elements interact with each other, whereas some act antagonistically with each other. So really what we need is a proper balance of nutrients in plants. There are 17 essential chemical elements for the essential growth of most plants. And of course, there's your little mnemonic device there, C. Hopkins Cafe, managed by my cousins, Mo, Nigel, and Clyde, to remember those essential elements. When a plant completely dries out, its dry weight is primarily composed of carbon, hydrogen, and oxygen. Those come from water and carbon dioxide. The remaining elements come from soil. So we got our primary macronutrients, our secondary macronutrients, and then, of course, our micronutrients, which are often referred to as trace elements. Know which categories those nutrients belong to. Also know that there is this law of the minimum, which basically just states that even if all other elements are present in sufficient quantities, that one deficient element will not allow the plant to grow and develop properly. Also know that mobility in the plant is also a very important factor in nutrition. Know that nitrogen is the largest primary macronutrient and is the most common limiting factor for plant growth, and generally needs to be supplied every season, and sometimes multiple times in a season. We often supply nitrogen through two forms, ammonium and nitrate. And there is a nitrogen cycle that is basically acts a lot like the carbon cycle to make these nutrients available to plants. We have nitrogen fixation, of course, which is the natural conversion through some types of bacteria of atmospheric gas into ammonia. And that occurs often through rhizobia bacteria, nitrogen fixing bacteria. And many of those occur naturally in legumes and some other species of plants too. Which creates this symbiotic relationship in which the plants supply the bacteria with a home as well as with some carbohydrates, and the bacteria supply the plant with ammonium. And they live there in these things called nodules in the roots of the plants. The Haber-Bosch process is something that we've created to basically convert the same process, basically, but it requires very high temperatures and pressure in order to be able to actually turn it into a nitrogen fertilizer bonded to a salt. In soil bacteria, in the rhizobia, the same thing happens, except it does not require a tremendous amount of pressure and can occur at room or soil temperature. Nitrogen availability, as well as other elements as well, occurs through mineralization and or ammonification, which is the conversion of that organic nitrogen to ammonium. And then the conversion of ammonium to nitrate through nitrification, which basically is the most available form of nitrogen for plants. Nitrate is an anion, though, and it can easily be leached from the soil. It's also very mobile, so that can create some other issues, such as nitrogen runoff into other ecosystems. Notice some of these deficiency symptoms, they're the really common ones anyway. Nitrogen, of course, is responsible for the dark green foliage in plants because of its presence in chlorophyll. And so one of the first signs that we see as nitrogen deficiency is the yellowing of lower leaves. Again, because nitrogen is mobile. So it will take it from the lower leaves and move it to newer growth in order to allow the plant to continue to grow. Phosphorus is important for several different reasons. It's often the second most limiting element for plant growth. Much of it is tied up in organic matter, and soil pH is very important for phosphorus uptake. So really we need between 6 and 8, ideally, kind of hovering around neutral, what many people call the sweet soil. Phosphorus can be taken up or helped to supply phosphorus to the plants by mycorrhizal fungi, which is basically just a fungus absorbing it from the soil and moving it into the plant in exchange for carbohydrates. And of course, mycorrhiza is available commercially. So what does phosphorus deficiency look like? Often, again, it's mobile. So the lower leaves can appear darker green than expected, and often a red or purplish color, often on the undersides of the leaves. Potassium is critical for the opening and closing of stomata. So for gas and water exchange. Because potassium is generally a cation, it's retained in the soil and not easily leached. And deficiency of potassium usually begins to appear as chlorosis and necrosis starting at the margins of leaves. And it is vein limited. So if you notice, the veins there are green, but it's this interveinal chlorosis, yellowing of leaves. No, calcium does. It's very similar to what it does for people. Very important for providing structure for tissues of plants, especially the cell wall. And generally, if there is a deficiency, it often appears almost like what looks like a rotting, but is actually just the tissues collapsing and becoming soft. It's just blossom end rot in tomatoes. However, that can be fixed through foliar applications. Magnesium is also an essential component of chlorophyll. So we get chlorosis with it as well. It's also important for energy transfer because it's a necessary component of ATP. We also generally get intraveinal chlorosis of lower leaves with magnesium deficiencies. Iron is very pH dependent, and it is an immobile element. So usually what we see for an iron deficiency is intraveinal chlorosis, but of the upper leaves rather than the lower leaves. And adjusting soil pH is often a pretty surefire way to prevent or correct iron deficiencies in plants. Whenever we're looking at diagnosing plant problems, we have to look at multiple measures for nutrient deficiencies because, one, those symptoms can vary from plant to plant. If we have more than a single element involved, then they can look different. They can also look like diseases or other disorders. So one way to kind of look at nutrient deficiencies and look at correcting them is to try to give a small amount of whatever element you suspect and then see if that corrects it. Also, we can perform tissue analysis. There are some tools that you can do that in the field. Of course, you can also send tissues into the lab, but that takes a long time. So, remobilization is basically just where the plants are able to reallocate those nutrient resources to other parts of the plant. Usually that's done for reproduction of the plant, so often they're moved to things like the flowers and seeds. And that is the completion of the Unit 2 review. Both of these are up on Blackboard, so you guys can see. Again, you can use this review during your tests. That's fine. I do want to note that when we get to the midterm, at this point we're like 3/4 term exam, that it's going to be proctored, so you're not going to be able to use these. So use these tests as a way to practice for that. Anybody have any questions, comments, or concerns?
You said this test isn't going to be proctored.
This test is not proctored. It will be on Blackboard, and it'll be available for about a week. It will be the exam that is proctored.
Okay.
When did you say this test was going to be available?
Starting Wednesday afternoon.
Okay.
And you said it's going to be up for a week?
Yep, for 1 week.
Okay.
Anybody else? All right. Well, you all have a great day. Finish up those quizzes so we can get those graded and have a little bit of quiz review and see if any of you struggled on anything on Wednesday.
Okay. Thank you.